Evolution of the immunoglobulin superfamily and the adaptive immune system, AIS. Brief overview. The AIS provides a quintessential example of shape-specific molecular interactions and binding events, SSM-IBE, Chapter 12. Gene duplication has combined with other potent sources of genetic diversity, including VDJ recombination and somatic hypermutation, to generate an unrivaled higher-level adaptation that effectively seeks out and destroys potential threats to multicellular individuals, while distinguishing foreign entities from the self. It does this by binding and internalizing antigens, which are often fragments of protein chains, and then presenting them to antigen-presenting cells. Then the antigens stimulate production of clones of individual T cells and B cells. These clones express specific T cell receptors and antibodies that bind to specific antigens with exquisite specificity. Often, B cells secrete soluble forms of the antibodies. Under the aegis of natural selection, the AIS has been expanded and fine-tuned so that it can effectively recognize, bind, and neutralize the diverse antigens characteristic of a vast number of different potential threats, including invading bacteria, viruses, toxins, and even cancer cells. As with all products of natural selection, these capabilities evolved over many generations from more primitive forebears. Mammals can produce an astronomical number of different antibodies, probably on the order of 1,000 billion different ones in humans. The AIS also generates a vast range of T cell receptor specificities. T cells include both helpers and cells that can kill other cells, designated CD4 and CD8, respectively. CD4 cells send signals that generate clones of numerous specific T cells and B cells that bind to particular antigens. These are called memory cells and often circulate in the body for years. When the antigen is encountered after this primary immune response, these cells can rapidly proliferate and effectively eliminate the threat before the threat gains a foothold. The purpose of a vaccine is to elicit a primary immune response so that individuals are prepared to effectively fight pathogens that might otherwise cause future infections. Today, our empirical understanding of the expression, cellular functions, and evolution of the various members of the immunoglobulin gene superfamily is extensive. Several empirical techniques, including DNA sequencing, databases, and computational tools for systematic comparisons of DNA, RNA, and protein sequences, monoclonal antibodies, and flow cytometry, are just some of the powerful tools that have been fruitfully applied to the subject. Evolution of molecular mechanisms that generate diversity in the AIS during T and B cell development. How did the diversity of the AIS evolve? During the molecular evolution of the AIS, tandem arrays of literally hundreds of genes, members of a large and diverse multigene family known as the immunoglobulin superfamily, ISF, evolved via extensive gene duplication. Numerous DNA and protein sequences of ISF family members were identified by Leroy Hood and colleagues in the 1980s. ISF family members play a variety of now well-understood roles in the cellular functions of the AIS. A second crucial molecular component of the AIS is VDJ recombination, a novel mechanism that is active during the development of T cells and B cells. VDJ recombination randomly shuffles and splices diverse members of several multigene families to generate a vast repertoire of random recombinant T cell receptors and antibodies that can bind to diverse antigens. This random shuffling is facilitated by a major generator of diversity known as recombination activating genes, RAG. RAG are thought to have evolved from transposable elements, TE. Recently, a living molecular fossil, thought to be the progenitor of RAG, was discovered by Shengfeng Huang and colleagues. The widespread activity of TE in molecular evolution was mentioned in a section previously. A third major molecular component of the AIS is a novel process that rapidly generates diversity among the recombined T cell receptor and antibody sequences during T and B cell development, a process known as somatic hypermutation.